Welcome to the workshop on RNA sequencing data analysis. So before I start the workshop, anytime anyone feels that it is going a little faster, you can let me know. You can say, you can unmute, you can raise your hand, you can unmute yourself and speak, or you can put your questions in the chat box also. Anytime you have any question, any doubt, please feel free to ask your doubts, questions. Make this session more interactive. Just don't think that I'm teaching and you are learning. So whatever question comes, feel free to ask. You can use WhatsApp group also to ask your questions. I am responsible for technical queries. Other admin queries, team will reply. And every day morning, you will get, next day morning, you will get the recordings, which is linked to the YouTube. So make sure that you have given your email ID by which you log into YouTube. All right, so let's start the session. So today we will be looking at some theoretical part, and we will also look at some more important things which are required for RNA sequencing data analysis or data curation. So let's start. So the transcriptomic analysis. What is transcriptome analysis? So we know that we have thousands of genes, and these genes will get expressed into some proteins or functions to perform certain functions. So intermediately, when the RNA gets converted into mRNA, our gene, because there is a process. So when genes transcribed, that is called as mRNA, and mRNA translate into a functional protein. So proteins and other stuff, though there are different methods for their analysis, but they are very short-lived, and being a messenger RNA intermediary product, because directly it is coming from your DNA and translating into your protein. So it is feasible for a researcher or for us to capture the mRNA. So if we do the mRNA analysis, we can predict the protein also, and we can go back and look into our DNA as well. So if we analyze the entire mRNA, which is transcribed by the genes, all the genes, the entire profiling of messenger RNA at a given time point is known as transcriptome analysis. And now you might have asked one question, why I said at a given time point? Because we know that this is the fact that all the genes won't transcribe at once. As and when they require to perform certain functions, they translate into proteins. So over the period of time, our transcriptome change. So say suppose at this time point, you analyze your transcriptome, maybe after 2 days, 3 days, or certain time period, your transcriptome might change, because after certain time point, some more genes might require to get expressed, which are not expressing at T0. So that's why always transcriptome analysis at a given time point. Not it is going to be it can be changed. So this is again one more challenge, that because the transcriptome change over the period of time, while DNA is a stable molecule, once you analyze it for the entire of your life, it is not going to change. It will remain the same, except the somatic DNA variants. If it is a germline, it is not going to change. So keeping that in mind, keeping that all these facts in the mind, we will be looking at how the transcriptome analysis is performed from the NGS data. So in this workshop, we will be looking at some introductory part where we will be looking at RNA sequencing. What are different type of RNAs are there which we can sequence? What are different applications of RNA sequencing analysis? How the samples are processed? Because our responsibility is for the data analysis, not the sample processing. But there are certain people, those who work in the lab as well as in the data analysis. So not only, even though if you are not working in the lab, but you are dealing with the data, so you should be aware how the data is being generated in the lab. Because if your data is not good, you can go back and say that, yes, something is going wrong while processing the sample. So please take care of these things so that the data will be of good quality and the analysis will be more simpler. Not exactly simpler, but will be easy to analyze, and you will get a good quality interpretation from that data. And lastly, how to do the data analysis from RNA-seq. So in this, we will be looking at different aspects of RNA sequencing, and depending on those aspects, we will design a workflow, what we are going to practice in this workshop for the RNA sequencing. Because RNA sequencing is not just the RNA sequencing, there are different parameters, there are different ways for the analysis. Because it is not a very long workshop, it is a short workshop. So the idea behind this workshop to give you a flavor how to do the RNA sequencing data analysis. So in general, but there is no end or there is no limit of the analysis. It can be very, very advanced level as well. So this particular workshop is designed for the people or the students. They would like to analyze their own data, a small project or their thesis data by their own. So after this workshop, you are able to analyze your data. Also, the platform which we use to teach the hands-on, it is not depending on multiple factors. So it will be easy for any person who doesn't even understand the computational biology or coding to how to analyze it. So this is a very, very independent project or the platform where you can analyze your own data. All right, so let's start with the introduction. So as I mentioned that the transcriptome is the collection of all RNA which are transcribed by an individual or a population of cell at a certain biological state. These studies which are focused on mRNA and coding. So now if we see what are the different type of RNAs are there. So usually RNA are of two types. One is coding, another is non-coding. So as the name itself suggests, coding and non-coding. So they code for proteins. And they do not code for any protein. But it does not mean that they do not code for any protein. So earlier there was a thought that because these are not the important part of the genome because they do not code for protein. These are the only important part because they code for the proteins. But now over the period of time, we understood that these non-coding part are the regulatory proteins. So they are the basically the regulators which regulates the coding of these genes. So this is also important part how to analyze it. And basically in the coding RNA, messenger RNA comes. In the non-coding part, all small RNA comes. So what are those small RNA? Like your miRNA, that is the microRNA, which are the part of your untranslated regions, that is 5' or 3' UTRs. Your siRNA, your all non-coding long non-coding RNA. So this is your link RNA, then your pi RNA. So like that, we have several type of RNA molecules, small molecules, and they all somehow they regulate the these mRNA. Some are enhancers, some are promoters, some are there are different types of regulatory things. So in this workshop, we are not going to concentrate on non-coding RNA. We will be mostly concentrating on the mRNA analysis. So that is basically the coding RNA. So we will be concentrating on this RNA analysis. So studies interpret gene functions. So basically this RNA interpret the gene function, gene structure, and reveals the molecular mechanism of a specific biological process and disease occurrence. So any change in this coding RNA from normal state that might lead to any changes in the biological process and will lead to the occurrence of any disease. That is what we say a disease state. So the idea behind for this analysis, we can make the comparison normal versus disease. If you are going to treat this disease, so then we can compare between the, so then this will become our normal group and the treated group. So if we would like to see the effect of different drugs, we can compare with the non-treated and treated groups. So there are different types of comparison. There are different applications which we will be looking at in a later part. So this can also discover unknown and rare transcripts accurately, identify variable cleavage sites and the coding SNP, because when we are saying messenger RNA, it has only the exons, and basically when it will code for some protein, it will code only some coding sequences will be there which will be coding. So even though they are the exons, some part will not code for in the protein. So that is also called as non-coding part, and which will code for exactly the proteins, that is called as CDS. CDS is the coding sequences. So we will have the excess of coding sequences. So we can also do the variant analysis, which is coming directly from the coding sequences. So that is the benefit why we are doing the RNA sequencing. The high throughput sequencing gets access to the almost all transcripts of specific tissues, cells in a certain state by comprehensive and rapid complementary DNA sequencing. So now what we are saying, because we are doing the RNA sequencing, so directly we are not sequencing the RNA. So what we are doing, we are isolating the RNA, converting into cDNA, because RNA is very short-lived molecule. The half-life of RNA is very small. So immediately it gets converted into cDNA, and this cDNA we are doing the sequencing. So by which indirectly we are sequencing the RNA, which becomes the basis as well as the starting point for the research of gene expression. So because this is expression, so that's why we also say it as the gene expression analysis. So the main objective of transcriptome research includes the classification of all transcriptional products, determination of transcription, structure of genes, the start sites, 5', 3' terminals, that is UTRs, splicing pattern, post-transcriptional modification, and the changes in the expression level of individual transcripts during the development, any development. So now what all the type of analysis which we can do from the transcriptome data? We can reveal the structure of the genes. We can look at the start size, that is our promoters. UTRs, splicing, from where the splicing happen, intron-exon junctions. So these are the intron-exon junctions. So any modification in the transcript or the number of transcripts because of the splice sites. So we can do this analysis as well. Post-transcriptional modification, because we will do the capping, poly-A tail, this and that. So that all comes in the post-transcriptional modification. And the changes in the expression levels. So say suppose in the normal, any genes get expressed at 10 copies are there, but maybe if the disease is there, the same gene might have the 100 times expression. So this kind of expression levels also we can analyze from the transcriptomic data. So as I told you that in this we will be looking at messenger RNA, and as there are different applications of mRNA analysis, so most important application is the differential gene expression analysis. So we will be concentrating on this, more on the differential gene expression analysis from the mRNA in different conditions. So the analysis is same, but the interpretation and how you look at these data, that is different. So this has been widely used in different research areas like medical, clinical research, and drug development. So the same application, gene expression analysis, you can use in a variety of applications. So that also we will be looking at how you can use the same application for different areas. So here, if you see, this is the how the DNA will be there, and the DNA is a double-stranded molecule. And when the transcription happens, as I mentioned, all the genes will not be transcribed at once. So wherever the genes are present in the DNA, whether it is here or somewhere else, so whatever the number and the different genes are getting expressed, that particular region of DNA will only open. The entire DNA will not open. So the double-stranded DNA will open and make it as a single-stranded DNA. So now we know that in DNA we have double-stranded, and these are the anti-parallel strands. So if this is one is called as plus strand, another is the minus strand. So if you look at this, so this will be 5' to 3', then this will be 5' to 3' end. Some genes will code from this strand, some genes will code from this strand. So any strand the genes will be coding, then it will transcribe the RNA from there, and the process is called transcription. And once this mRNA, so this is our mRNA. When this mRNA forms, this will be released from the DNA, and DNA will rewind again, and this transcribed RNA will code for your polypeptide chain. And this process is known as translation. So here we are not talking about this process at all. We will be talking only about this. So now you know that in the gene, we will have all exon, introns, and when the transcription happens, so first step, everything will be there. But what will happen when the modification happens? So in the gene, we have all exons, introns, and when the mRNA forms, these introns will be chopped off, and these exons will join together. And then we have here 5' UTRs, 3' UTRs. So this again depends what type of analysis you wish to do it. And these are the splice junctions where these exons joins. So now say suppose when the transcript forms, one transcript can form from here, one can form from here, one can form from here. So these are the CDS sequences. So these are not translating. So these are the non-CDS sequences. So in these two transcripts, these are the non-CDS sequences. So that's why we are saying we will have the exact coding sequences from where the protein will sequence. So now when we are having the mRNA, we do the fragmentation into small pieces, and these pieces will be sequenced. So we will see how we do the fragmentation, how we sequence that. But here just understand, we fragment these because the transcripts will be larger, and we have two types of platform, sequencing platform. So one is the short read platforms. Another is the long read platforms. So short read means short fragments will be sequenced. Long read means long fragments will be sequenced. So usually they are in the KB, not even KB, up to just 600, 700 base pair. And these will be in KBs. So 10 KB, 20 KB, or longer than that. So here we will not talk about long read. We will be talking about the short read process, because mostly the RNA sequencing is performed on the long read, until unless we are not building up the transcriptome, reference transcriptome. Otherwise, for a normal process where the reference genome is available, short reads are good enough to do the gene expression analysis. Okay, coming back here. So after the fragmentation, once these reads will be sequenced, they will be aligned to the reference genome, and we count these reads, how many reads are aligned to each exon. So this is our gene 1, this is our gene 2, and you can see in this gene we have 3 exons, and here we have 2 exons. So these are the exonic part where our reads only will align, because they are coming from the exons, not from the introns. These are the introns. This is basically the intergenic region, because this is the region between 2 genes. Right? So this is how we do that analysis, but we will discuss all these things in detail. So basically there are 3 types of RNA based on their functions: ribosomal RNA, transfer RNA, and the messenger RNA. And out of that, because the ribosomal RNA just provide the space for the synthesis, transfer RNA carries the specific messages or the amino acid, and mRNA is the molecule which is coded from the genes. So here, that's why we just get the sequencing done for mRNA, and that is what we are going to analyze it. And if we look at the different type of, based on their functions, they are of different types, like small RNA and the large RNA, that is the transcript, that is the mRNA. In a small, they are the regulators, and mRNA is the coding RNA. So we are just looking at the coding RNA over here. Sorry. So now when we look at the application of the transcriptome analysis, so whether we talk about the biomarkers, tumor heterogeneity, drug resistance, or any of these applications, so for all these applications, the basis is the differential gene expression analysis. So if you understand what is the differential gene expression analysis, you can use this particular application in another applications as well. So let us try to understand first what is the differential gene expression analysis and how this analysis can be used in biomarker discovery, tumor heterogeneity, drug resistance, and another applications. So let's see what is the differential gene expression analysis. So let's take one example. So as the name itself suggests, so we are here looking the gene expression analysis between 2 groups. So that is the differential. So let's take some application. So how you can use them? Say suppose you have a rose plant. We know that we have different colors. So we would like to see what are the genes associated with the color. So that if you need more color or that specific color, you can transfer this gene and make a hybrid. Also, there are some species of rose which has a smaller flower, which has bigger flower. So anything you can do. All right. So what we are doing? So say take white flowers, take red flowers, take yellow flowers, and any other flower, color flower, keeping other factors normal. So if these flowers are white, red, yellow, but the flowers should be small, right? So we are isolating RNA from here. then converting into cDNA, and generating the data. So now what will happen? We have some genes, which we know because this is the plant and we know. So say suppose we have here gene 1, gene 2, gene 3, gene 4, and so on. So what we can do here, so we count how many reads are aligned from each data for these genes. Say suppose in this 10 reads are aligned, in this 15 reads are aligned, in this 30 reads are aligned. So like that, we can just denote something. This is all 10, this is 10, this is a 12, this is 25, this is 25, 30, and this is 10. Like that. So now if we look at this matrix, we can see and say that, okay, this is 10, so this is 15 and 30. So simply by looking at this matrix, we can say, okay, this gene is expressed in this and this. And as compared to this, this is high expression. So that's why which is responsible for yellow color. But whether this change is significant or not, that also does matter. If we calculate and put the statistical differences, that yes, this is statistically significantly different from this and from this, then they are differentially expressed genes. This is definitely not going to change because it is all 10. Now, if we look at here, this is fine. This is totally very high. But this change is statistically significantly different or not. So that all kind of analysis from this matrix is called as differentially expressed genes. So we can correlate, okay, in this particular matrix, if we see this is high in red, this is low in yellow. So we can say that, okay, this particular gene is responsible to provide the red and white color. But apart from this is also like high in this. So these two together might give you the red color. And this is only responsible for white color. And similarly, here you can see these two genes are responsible for yellow color. So like that, if we have this, we can make sure that what is responsible. If we look at in this, so this might be the common things. So it is also possible that only this gives you the red color and only this gives you the yellow color. So that is the idea behind differentially looking at these DGE, which genes are responsible for the process or for any kind of inference which we would like to take it. So how and where you can use? So one example I already gave you. Say suppose you want to understand if you have a plant, you have root, you have stem, you have leaves, and so on. So you have a different tissues. So if you would like to do the, would like to know that what genes are expressed in roots, stem, and leaves, that also you can do, because the roots perform a certain set of functions, stem performs different functions. So you can correlate based on these genes why this stem performs that function, because only those genes are expressed in stem, not in root. So like that, we can utilize the same application. So now if you take the disease condition, so one example already I have given you, normal, when the tissues are normal and it is disease. Even in the disease group, especially in oncology or tumors, you can segregate the disease group as per the stage. So you know that all the cancers, they have these stages. So you can segregate stage 1, 2, 3, 4, depending on the tumor type. because this might be the primary stage and this might be the advanced stage of your tumor. So some genes might expressed here, they might not be present here, or they might be highly upregulated at this stage. Some genes might be absent here, some genes might present over here. So all that changes can provide you an inference that how the disease progression happen. So you can utilize in the disease progression. Now you can see these genes are present here, but they are absent here. So because you can utilize these marker for your diagnosis. So you can make them as diagnostic marker. And over the period of time, if you see this is maybe the low expression, this is the high expression. So again, you can use that is in the monitoring of the disease. Also, so this is diagnostic marker. This is what you call it as a biomarkers. So biomarkers can be for the diagnostic purpose, for monitoring of the disease, for disease progression. And if you see these changes, which is very particular and prominent, the same gene you can use for drug development based on in which process they involved. So the same thing you can use for the drug development, drug discovery. It means that the target identification. So again, when you are identifying the targets, this can be for diagnostic purpose, for monitoring purpose, for drug discovery. So you can see within the same application with different kind of data, we can utilize for different purposes. If now take the other example, say suppose you have a tumor microenvironment. So in tumor microenvironment, we will have a different type of cells. So as you can see, we have different type of cells. Because these blue color cells will have a different transcriptome. Green color cells will have another transcriptome. So maybe I can just put T1, T2. And the green color cells might have a different transcriptome. So they will have some common genes. They might have some different genes. So if we do the analysis, we will get to know how many different type of cells are present in this microenvironment. So this is all again based on the differentially expressed genes. Now, once we have the different type of cells, because they are coming from different cells. Right? So we know that how to treat this particular tumor microenvironment. Because we want to kill these cells, all the cells. But what happens? Say suppose if you are using drug 1, which can effectively kill these green cells, but not effectively can kill red and blue cells. So what will happen? Still your blue cells will be there and your red cells will be there. Only the green cells will be killed. But these are also the tumor cells. So if in this case or in this scenario, other cells will multiply faster than the before. So this will make your tumor progression faster. Or because this drug is least effective, less effective than the green cells, so some cells might kill, some cells will be there. So after the treatment, these cells might again proliferate and give you the tumor. So this is called as relapse. So based on the differential gene expression analysis, all these can be done together. In this scenario, there are two types of analysis can be done. One is the bulk RNA-seq. Another is the single cell RNA-seq. So what is bulk RNA-seq? If you take the entire tumor and then do the RNA isolation and type it, that is called as bulk RNA-seq. What will happen in this case? What is the drawback of that? This is very easy. And if you just would like to do something, this is easy to perform. But what is the drawback? Say suppose these cells are having less in number. Because you are taking the random tissue, you might be able to capture it or not. So if you dissociate this tumor into single cells and you isolate RNA differently from each cell and analyze it. So what will happen? We know from which cell our transcriptome is coming and we can compare. Maybe these 4 cells might give you the same transcriptome and these 2 cells might give you a different transcriptome. So if you compare these cells, you will get to know exactly from where your transcripts are coming, where your genes are expressed. So, but this is really a very huge data it will generate, because say suppose if you are generating from 3000 cells, it will be like your 3000 samples, because every sample treats as a different sample. So in this workshop, we will be also dealing with only bulk RNA-seq, not the single cell RNA-seq. So what is the take home till now? So we will be analyzing the mRNA. We will be analyzing the short read platform or short read data. And we will be dealing with the bulk RNA-seq data. And we will be concentrated on differentially expressed gene. That is the gene expression analysis. And in most of the cases, RNA-seq deals with the cancer data. This is very, very helpful in cancer genome, or you are treating with some drug, anything, or developmental stages. So that is important application. So say suppose now you would like to see how the zebrafish, because zebrafish mouse, they are the model organisms for human analysis. So every time you do something or develop some drug development or anything, you cannot directly go for human trials. So you have to have some model which is very close to human. So you can test on them, any gene modification or anything. So these are the model organisms. So it is easy to replicate the human physiology and the genetics in these model organisms. Then what is important here? So when we do the RNA-seq, because we would like to see the gene expression analysis, so we have to align our data to reference genome. So if we see the RNA-seq, broadly there are 2 applications. So one is the building the reference transcriptome, and the gene expression analysis. So here we are not going to build any reference transcriptome. We will be looking at gene expression analysis. So we will be only looking the reference based analysis. So this point also you can add it over here. So we will be looking at reference based analysis where the genome's reference genome is available. We will be looking at mRNA, short read data, bulk RNA-seq, and the differentially expressed genes. So these steps we will be looking in detail how the data is being generated. So if we go back, so tumor heterogeneity, drug resistance. Already I told you, because if the drug can kill only one type of cells, another cells will, and when you expose any cells with a small amount of some drug, they will get the resistance if they are not responsive. So if we understand the tumor heterogeneity, we can give the combination of drugs, and then we can also deal with the resistance, drug resistance for the tumors. So this is how you can, all these application, novel molecular signatures. So if you compare the different states, different time points, then you might come up with some novel molecular signatures which are not present earlier. Now they are present. Regulatory proteins. So if you will analyze the non-coding RNA, then you will get the analysis of regulatory proteins. So again, it is the same thing. So when you analyze the regulators and the transcriptome together, you know that what the genes are getting expressed and what are their regulators. Unknown subtypes of immune infiltrating cells. So again, we know that there are different immune cells like T cells, B cells, NK cells. So if we profile on the single cell or the gene expression, we will know what different type of cells are there. So for some organisms like human and mouse, we already have the known immune markers which are helpful for the cell typing. But for some animals or some organisms, we still do not have the immune type signature for the gene markers. So their single cell is important to build such kind of markers. So it is a combination how you look at your data and what needs to be done. So if something is unknown, you can do the discovery using a single cell and then come back and then look in the bigger population, because the single cell is costly as well as the analysis is very complex as compared to RNA-seq. But you need to have some information when you do the bulk RNA-seq. You will get the all these video recordings. So what all you can do the differential development stages analysis using the same application, cell types, differentially expressed genes in different cell types, different species, different treatments, different tissues, different genotypes. So all that can be done. But if you look at the like the data analysis of your small RNA and the mRNA, they are very similar, just how you infer and where to align, that is only the difference. But at the same time, first is the mapping to the reference genome. In case of RNA, we get the genes and the where on the exons they align, while on the small RNA, they will align on the non-coding part. So once we see, compare their transcription levels, that is the expression levels, then the comparison with the gene expression, mRNA expression, and then do the downstream analysis for their ontology, their functions, their biological functions, their target genes, what the mRNAs are upregulated, downregulated, and then there are certain downstream analysis which can be done on the advanced analysis. So there are different platforms, as I mentioned to you. So they can be categorized into basically two, a long read and short read. So like Nanopore and other platforms are the long read platforms, while Illumina is the most commonly used platform as a short read platform. So here we will be dealing with the Illumina data. And usually the Illumina data is in the FASTQ format. We will be looking at different file formats which will be generated during the process and how the data is being generated. And usually the RNA-seq can be performed by two methods. Not RNA-seq, but the gene expression analysis can be performed in two ways. One is the using microarray and another is the RNA sequencing. So when initially the gene expression analysis is started, it is started with microarray platform, and then over the period of time, RNA-seq took over the gene expression analysis. So there are benefits of both the platforms and there are drawbacks of the platform. So if we look at the microarray, because it is a hybridization based platform, so we need more amount of RNA for the gene expression analysis. While RNA-seq, you need a little less amount of sample, which gives you the entire transcriptome. In microarray, because it is a hybridization based platform, so we need to design the probes for hybridization. And to design the probes, we need to know the genes, what the transcripts are translating. So this will give you only the known genes transcription or the expression level. Unknown it will not give you. But in RNA-seq, because it is independent of all these factors, because there we are not using any primers and probes, so we are able to capture the novel gene expressions as well. So that is known as novel transcripts also we are able to do it. Any deviation in the known gene expression of the because of the splice, that also we will miss in the microarray platform. But in RNA-seq, we will be able to capture that process as well. In microarray, you will get only the gene expression levels. You will not be able to get the SNPs from the coding sequences. But in RNA-seq, along with the gene expression levels, you can analyze the variant analysis also from the coding sequences. So there are many advantages over microarray from the RNA-seq. Also, RNA-seq data can also be used for the alternative splicing, novel transcripts, and the fusion genes. They are not possible from the microarray data. So you have to see whether you want to just go for the known gene expression or some novel expression discovery. So based on that, you can choose to go either for microarray or RNA-seq. But if you know what the genes you are looking for the gene expression and you want to screen for large number of samples, you can use microarray. But if you are not sure what you have to look, then go for the RNA-seq. Also, when you are doing the microarray, it is based on the intensity. So sometimes the intensities varies and always you have to run the control. In RNA-seq, you really do not need the control to run it. There are various ways how you can make your controls. But because in the microarray, it depends on this intensity, so it is mandatory to run the control with the known probes, so that you know what the intensity they will provide you. Then only you will be able to compare with your sample. So these are the pros and cons of microarray versus RNA-seq. So as I mentioned, there are two types of RNA-seq analysis. One is the alignment based when the reference genome is available. So that experiment is called as resequencing, and which is characterized by the availability of well-characterized reference genome and annotation. And if you are doing such kind of experiments, usually the data is generated at 25 to 30x and the read length is 2 into 75 base pair. So what is the meaning of 25 to 30x? All the exons, your read data at least should have the 25 to 30 reads minimum covering all the exons. It can be more, it can be little less, but not less than this. So every exon is sequenced this many times. De novo, when the well-characterized reference genome and annotations are absent, and this involves the building of a transcriptome, and generally the reads are sequenced at the depth of 50 to 100 because we need more data, and the bigger read length, that is 2 into 100 base pair. So these are the general guidelines, but depending on the type of analysis you do, you have to modify these parameters while generating the data. Say suppose if you have a more variability in your genome, but still you would like to sequence, like you have a resequencing experiment, then you can increase the read length and sequence has more depth. If you want to capture your lowly expressed genes or whatever it is, if you want to go for the translocations and other things, you need to change your depth and as well as the read length. Because if you are going to analyze the translocations, you need longer reads to be there. So this is based on the type of analysis you want. You have to take care of these data generation parameters. So basically, you have to take care of the depth as well as the read length. This is the general workflow. First is the RNA sequencing. Before that, first the isolation, converting to cDNA library preparation, which we will be looking in some time. But once the RNA libraries are ready, they will be sequenced and it will generate a raw data. This data, when we get the raw data, we will be looking at the QC because it is important to look at the data quality. Data quality need to be good because if the data quality is not good, the interpretation and analysis will be not accurate. So once we are sure, then the read count will be performed, and depending on the how many samples, control and other things are there, then the normalization, different plots, statistical analysis, visualization to see what is the variability between the sample, within the sample group, what are the genes upregulated. So this is called as heat map. This is called as volcano plot. So usually heat map and volcano plots are used to see what genes are expressed or downregulated. So once we have that, we can do the gene ontology, look at the biological process, cellular component and molecular functions. We can also look for the gene-gene interaction, and some more advanced analysis can be done on the evolution. So how the proteins your genes are getting in over the period of evolution, how they are changing. So this is the general workflow which we will be looking at. So we will not look at the evolutionary analysis because this is highly advanced analysis, but other analysis we will be looking at in the gene expression analysis. So this is the sample processing. Let us start with the sample processing, how the sample processing takes place. So first we have to see the RNA isolation. So there are different, so first you have to select the source from where you are going to isolate your RNA. Based on source, you can choose your method of RNA isolation. So well-validated methods are there. If nothing is available, depending on your source, you have to validate your method by reading your literature, what is available, what is not available, what the reagents available in your lab, all that you have to see. So at this step itself, there are two things you can do. As I mentioned, that we can do the small RNA analysis or the mRNA analysis. So when you are doing the RNA isolation, if you want to do these analysis, if you want to do only mRNA, you can isolate only mRNA. If you want to do only small RNA, at this step itself, you can isolate small RNA. Or if you do not want at this step, you can isolate the total RNA. After that also, you can do this. So if we have the total RNA, This we have to do the quality control on that, and basically two types of quality control. So quality and the quantity. So there are different instruments to do that, but usually the quality is measured in terms of RIN number, which should be greater than or equal to 8.0. Ideal quantity is 30 nanograms per microliter. It can be more. If it is more, you can dilute. If it is less, you cannot concentrate, or you can just try to do the reprecipitate and concentrate it. But it is really very tough to do that. So there are protocols available with the low amount of your sample. So that all you need to see, because if you are dealing with FFP samples, you cannot match with this quality control. It will be, RIN number will be little lesser, or the quantity also will be less. So depend on what source you are choosing it. So if you have RNA, you want to enrich for your, so first immediately convert into cDNA. Then we know that in the total RNA, more than 80% of the fraction will be ribosomal RNA, which we have to remove. So there are two methods to do that: enrichment of mRNA or depletion of ribosomal RNA. So this is also known as ribo minus or ribo zero. Here in the enrichment, we know that it is a poly A tail. So mRNA will have the poly A tail. So using the oligo DT, you can enrich your mRNA only and convert into cDNA. So once we have this mRNA, because this is the mRNA and small RNA. So library preparation is almost similar. First, for anything, you have to convert this. At this stage also, you can pass through different columns so that they are size specific. So it can separate your small RNA and mRNA. You can use beads also. You can use columns also. There are many things available for this. But whatever it is, because these are small, so here we are not doing fragmentation. So no fragmentation. Because these are the transcripts, they are the large transcripts. So fragmentation. So usually there are two types of fragmentation: physical and enzymatic. So we are not using restriction enzymes, no RE. Because usually the restriction enzyme cuts the DNA at a specific site. So that is not our aim. So whatever the method you choose, our aim is to get the size specific. So controlling the temperature, there are specific enzymes available for the same process, or ultrasonic waves can be passed using for the physical methods. So whatever the method you choose, by controlling the temperature, then the frequency of the waves, and the time to pass, this can control your size. So once the size specific fragmentation is there, the next step is the end repair and adapter ligation. Because these are, so cDNA, when you convert your RNA into cDNA, that will be your double stranded. So it will cut in at a specific size. So you have to repair the ends. So these are the blunt ends. So this will give you the and repair. And what are the adapters? Usually they are the 8 to 10 base pair synthetic oligos. Because when we cut, we do not know where it is coming and what is the end sequences. So when we put the adapters here, because these are the synthetic oligos, we know what these sequences are. So these are our adapters. Usually these adapter sequences are taken for Illumina platform. Usually they are taken from P5, P7 promoters. So what are the promoters? Where the transcription starts. So these are the promoter sequences P5, P7, and this is also known as universal adapters. Illumina universal adapters. So after this, once this process is done, the next step is the bar coding and indexing. So these are also 8 to 10 base pair of synthetic oligonucleotides, which will be attached to these fragments. So why we are attaching now the indexes or barcodes? Because NGS can generate more amount of data, so we can pool many samples together. So if we pool the samples like this, because all the fragments from different samples at this stage will be same. So later on, we won't know which fragment is coming from which sample. And we want to know which fragment is coming from which sample. So we attach some specific sequences so that we know from which sample these fragments are coming. So these are specific to your samples. So two samples cannot have the same barcodes. If you are running in the same run. Same run or pool. You can have the same barcodes, but then you have to make a different pool and run in different lanes or different chips. So now once you have this, here you have the index sequences. And usually these index sequences are I5, I7. So these are different sequences. There are a set of for Illumina. They have designed these indexes, which you can make the combination. So if I5 is here, I7 is here, this will be I5, this will be I7. So like that, for one sample, you are putting the one set of indexes. For another sample, the different set of indexes will be there. Yes. So what is happening in the barcodes? So say suppose this is my one sample. This is my another sample. So I am doing the fragmentation. I am putting the adapters. Same adapters I am putting because they are just two adapters P5, P7. So they will be same in all the sample and this is in the different reaction. So this is my reaction 1, this is my reaction 2. Now after this step, once the library amplification is done, one step is the pooling. Here we have to pool different samples, S1, S2, S3. So this can come from the same library like RNA library or any other application. Now if you add the same thing, same indexes, it cannot differentiate between this fragment and this fragment because they are same. We cannot differentiate from where these fragments are coming. So that's why we add the indexes so that we know from where from which sample the indexes are coming. Say suppose I have added index 1 to my sample 1, index 2 to my sample. So based on this sequence, I can segregate these fragments. If I will add I1 here, all the fragments will be same. I cannot segregate these fragments from each other. So that's why we add the different barcodes. You can understand when you go to a mall, every article will be barcoded and all the information is saved in that barcode. So you just read the barcode with the barcode reader and then it will give you the entire information. So this is also same, but these barcodes are 8 to 10 base pair oligos. Understood? Is this clear?
Yes, ma'am. Yes, ma'am.
So barcoding and indexing. Then the next step is the library amplification. So why we are doing library amplification? There are purpose for that. So whatever the sample you are taking, it will have many cells in it. Right? So when we are doing the isolation of RNA, my RNA will come from here, will come from here, will come from here, will come from here. Isn't it? And they will come from here as well. So what is happening? My RNA molecules are more here, little less here, and very less here. When we do the fragmentation, more fragments will be here. Same less fragments will be here. Very less fragments will be here. I want to capture this. So what will happen when if without any amplification, if I will put for sequencing, you know it is all competitive PCRs. Whatever more in number, they will be sequenced more. I want to get captured these cells. So that is the idea of getting the library amplification so that these fragments will be enriched and get the sufficient number so that they will also be captured along with these fragments. So because these are the less in number or maybe low expression. So that's why that is the benefit of RNA-seq. We will be able to capture lowly expressed genes as well. Right? So that's why we are doing this library amplification, and it is generally up to 8 to 10 PCR cycle. These are sufficient to increase these fragment numbers. But if we do usual 30 to 40 cycles, after this cycle, only duplicates will form. No new amplification will be there. So that's why here we are doing only 8 to 10 cycle PCR, not the regular PCR. Once this PCR is done, my libraries are ready. And for this library also, we have to do the QC. Before pooling, we have to do QC. Again, for the quality and quantity. Because when you are pooling, it needs to be in the equimolar concentration. Quality, because you have done the fragmentation, say suppose you have done the 100 base pair fragmentation, plus you are adding both the sides your adapters, so 10 base pair, 20 base pair on each side. You are adding the indexes 10 base pair on each side, so 20. So ideally it should be in the range of 140 to 150 base pair your library size. But if you see on the higher side or on the lower side, it means that your fragmentation or any ligation did not work. Basically, if you are looking at here, your fragmentation did not work. If you are looking a lower side, your ligations did not work. And if you are seeing this size at a smaller size, maybe 40-50 base pairs as well, if you see additional peak over here, these are your only adapters, nothing else. So that's why you have to do all these QCs and get rid of adapters here. Sometimes you are not able to get rid of this. So when we do the data QC, we also look whether adapters are there or not. There also we do the cleaning for adapters. Once the library QC is done, then pooling happens. And after pooling all the samples in one reaction, it will go for the sequencing. So in Illumina platform, the sequencing happens in the flow cell. So this is the flow cell. In the flow cell, you will have the lanes. So this lane looks like this. And each lane is divided into small squares. And this small square is known as tile. If you look at the bigger picture of this tile, this is like this. And on this floor, the same adapters P5, P7 are attached. If you look at the close view of this, it will be like this. So this is P5, this is P7, P5, P7. Like that it will be. When you are doing the sequencing, you can do in two modes. So this is your fragment. You can sequence in both the direction. That is called as paired end sequencing. You can only read in one direction. So this is called as single end sequencing. So if I draw this nicely, how it will be? And if you remember, what is the structure of your fragment? So this is your adapters. So this is P5, P7. This is P5, this is P7. Here your indexes are there. So this is I5, I7, I5, I7, right? So this is how your fragment will be there. So basically, this is your 5' and 3'. This is 5', 3'. So this 5', 5, 7, they are complementary to each other. So this is 5, 7. So your fragment will come and attach here, because these are the complementary. And before you put in the sequencing, you have to denature so that it will be single stranded. So this is P7. So the other end will be P5. Now, if it is a paired end sequencing, because we are sequencing in the both the direction. So this end will come and attach to this nearby P7. So this is P5, this is P7, this is P5. Now this is P7, this is P5, right? So your fragment can sequence in this direction also, in this direction as well. So complementary base will come and attach. So this fragment is growing. This is how the sequencing happens. This structure is known as bridge. So that's why it is known as bridge amplification. because this amplification is happening over the bridge, across the bridge. That's why it is known as bridge amplification. The sequencing is happening because the chain is growing. That's why it is known as sequencing by synthesis. We have millions of tiles in each lane. In each tile, we have millions of such spots. And on each spot, this process goes on simultaneously in a parallel way. So that's why it is known as massively parallel sequencing. So these are the three features: sequencing by synthesis, bridge amplification, massively parallel sequencing. So now whenever any base is attached, fluorescence will emit and that fluorescence will be captured. And this will, because this is so highly structured process, because this is happening at millions of spots together. So millions of fluorescence at one time it will be emit and which will be captured. And this will produce an image. So the first data is produced in the form of image and it is stored in the BCL files. Then this image gets converted into sequence file. So that's why it is called as base call process. We know that we have only ATGC, so only 4 colors. So these colors helps in identifying the bases and calling the bases. Now, because it is just the random process of the fragments, any fragment can go and attach anywhere. So in these files, we have all the sequences from all the samples. So we have to segregate these sequencing of samples to individual samples. So this process is called as demultiplexing. If the sequencing is done in paired end mode, you will get 2 files which will be having the notation either _1, _2 or _R1, _R2. If you have performed only single end, we will get only 1 file. And these files usually generate in the format of FASTQ format. It is a 4-line format. The first line starts with the @ symbol and known as sequence header. So this will have your unique sample name or ID, flow cell information, machine information, adapter, barcodes, all these information. And at the end of the line, it will have your read number 1, 2, 3, 4, 5. And if it is a forward file, it will end with _1 or _R1. So this is for the forward file. If it is a reverse file, it will end with _2 or _R2. So this is your reverse. So looking at this, you can identify which one is forward file, which one is your reverse file. Second line is your sequence line. So this is our sequence line. The third line starts with the plus symbol, and this is known as quality header. So in this, the information is same as here. So other than the plus symbol, you cannot see anything because these are huge files. So nothing will be written in this line except the plus symbol. So looking at plus symbol only, you can identify this is the quality header. The fourth line will have the quality score for each base, and usually it is written in the special characters, which is known as ASCII letters. And when we do the QC, these letters get converted into a numerical score, and this is represented as capital Q, which is greater than or equal to 30. So this is known as overall data quality score. So now you can ask one question, why this 30? So let us see why this is set at 30. So if we say Q is equal to 10, this means that one base pair error in 10 bases. So which means my process is 90% accurate. If I say Q is equal to 20, one base pair error in 100 base pair. 99% accuracy. If I say Q is equal to 30, so one base pair error in 1000 bases. 99.9% accuracy. If I say Q is equal to 40, one base pair error in 10,000 base pairs. 99.99% accuracy. If you keep going on and calculating, then also theoretically it will never reach to 100%. It will give you only this. Because no process is 100% accurate, some error will be there. And why 30? Because these are the short read sequences. We are maximum sequencing up to 600 to 700 base pair. And this particular error, one base pair error in 1000 bases. So we can assume that our error is negligible over here. So we can consider this as our standard. More is fine, but if you are going to reach to 40, then you have to sequence 10,000 bases, which will be very costly and very difficult to sequence or achieve. This is easy to achieve. So that's why we set it as 30. If your sequencing is lesser than 100 base pair or somewhere between, you can even set your quality is greater than 25 or so. Depends on the application and other stuff. But this is ideal to achieve, easy to achieve and achievable. So that's why we set at Q is equal to or greater than 30. So today I will stop here. If you have any questions from here, you can ask. Any questions? All right. If we do not have any questions, I will stop the session here and we will see you tomorrow at the same time. Thank you.